In this tutorial, I would like to introduce you how can we apply tools from network neuroscience and graph theory to brain imaging. I will specifically focus on two types of analysis called graph Laplacian and graph convolutional networks. Here is an outline of the talk today. First, I will have a brief introduction of how can we start with constructing a brain graph. And once we have the graph fixed and what kind of analysis we can apply for imaging data, for instance, the graph Laplacian, and one more step forward is to build a deep neural networks on brain graphs. And I will also show you one case study of using the GCN, the tools called GCN, to do brain decoding. And last, I will I would like you to practice on the notebooks along with this talk. So first, what's a brain graph? So the graph has two key notions. One is the node, and the second is the edges. So in brain imaging data, the node was first constructed by with various types of brain atlas, and the edges could be defined by different types of brain connectivity. And once the graph is fixed, there is various types of analysis we can do. For instance, the shortest path and the clustering efficiency and the global efficiency and extra and the modularity analysis and extra. So what are exactly the steps in order to get one brain graph? So the first one is to define the node. In the literature, there is various types of tools that you can use. For instance, the Brodmann Atlas, which is actually based on the cytoarchitecture, and the Brain-Network Atlas, which is based on the diffusion tractography, and the Yu's famous atlas, functional atlas, that was defined based on the functional connectivity, and as well as the Glasser Atlas that has merged multimodal information into one combined atlas. So this figure actually show you how many different modalities of imaging data you can use. Right now, in the current literature, the most widely used modalities are the diffusion and functional MR. Once you have the node defined, then then we need to know how the nodes are connected to each other, which is actually calculated with various types of brain connectome. For instance, we can use a diffusion tractography, or we can calculate resting state functional connectivity by simply doing a correlation between the time series of the two brain regions. And after that, we already have a brain graph, and we can start doing more networks-based analysis. And here, I would like to introduce. I would like start with the introduction of the notions in graph theory. For instance, the adjacency matrix, which is actually indicate whether the two nodes are connected or not. And based on it, we can define the Laplacian matrix and normalized version. In the bottom, I actually show a simple example of a simple graph with only 6 nodes. So what is first, based on that, we can define the adjacency matrix. For instance, the node 1 was connected to node 2 and 5. So in the adjacency matrix, we can see in the first row, only the second item and the fifth item are marked as 1, and the rest are 0. And then once we have adjacent matrix, we can define the degree matrix, which is actually the sum of each row, or we can see how many edges for each node. And at last, we have a Laplacian matrix by simply minus the degree matrix from the adjacent minus the adjacent matrix from the degree matrix. It's quite simple, right? And so the next step is how can we use such simple, like elegant version of the brain model, elegant version of the modeling of the brain. I will start with a simple tool called graph Laplacian. And the key notion of the key step of graph Laplacian is actually to do a spatial decomposition, which will give us two sets of variables. One is called eigenvalues, as marked as lambda here. Another set of variable is called eigenvector, which is marked as v here. So what does these values mean? What does these two variables means? So here I show you a simple example from natural images. So on the right, first we have like 3 examples of the images, and so what does the eigenvector looks like? So we can see like starting from the second vector to the first vector. The eigenvectors actually capture different patterns inside of the image, and each pattern corresponding to different frequency. So the frequency means how each pixel was different from its neighbors. So if the difference is huge, it means this is so we can see in the second vector, it's comparing to the fourth vector, it's more smooth. Here I didn't show the first eigenvector because we know like the first eigenvector will be just the average of the entire image. So with this tool, how can we apply to brain imaging? The first simple example would be to do brain pixelation. Actually, this tool has been widely used in brain pixelation. Starting from 2004, Johansen Berg has already used the second eigenvector to do reordering to separate SMA and pre-SMA by simply reorder the second eigenvector. And in one of my previous study, we use the tool called spatial clustering, which is actually apply a k-means clustering based on the first k eigenvectors to separate different components inside of the substantial diagram and we were able to show each subdivision actually show significant different connectivity pattern. The same idea have been used to construct the whole brain to construct a whole brain atlas called brain atom atlas, and the brain atom atlas not only provide you just the atlas itself, atlas of 200 region, the atlas of 200 regions. It also show you how each region come with a distinct connectivity pattern. So beside the pixelation, we could also use graph Laplacian to understand how the brain was organized. The first example is from Daniel Margulis. He has used the eigenvectors from the Laplacian matrix that was constructed based on the diffusion spectrography, and he was showing, and there were mainly the main connectivity gradient in the whole brain. The first gradient was actually from the primary cortex to more high order cognitive function, for instance, the frontal parietal networks and the DMN and default mode networks. And the second gradient was actually switching from visual to ranging from visual to motor functions. And the second example is instead of is directly apply multiple eigenvector is to actually directly mapping the eigenvectors to the whole brain, which actually give you different functional networks, eigenvectors of the functional connectivity brain, a functional connectivity matrix. which will actually give you different functional networks. So far I have introduced you the simple but very powerful tool by using graph Laplacian. So next, since this is a deep learning tutorial, I would like so we will introduce you how can we use this technology and merge with deep learning architecture. So here we start with, and here we have this very nice tool called graph convolution networks. So one step, so I would remind you again of the graph Laplacian on the red box here. So one step from the graph Laplacian to graph convolution neural networks is to start with graph, is called graph Fourier transform. So one step towards the GCN is to one step towards the GCN is called graph Fourier transform. So first, in this case, we don't actually just have one value on each node of the graph, but we have a more dynamical graph, which means at each node we have a time series. And what does the graph Fourier transform do? So as you can see, so as introduced from this image, we start from graph signal on the left, and then we apply the graph Fourier transform that was basically transformed the signal, the original signal defined on the graph to a spectral domain by using the entire set of eigenvectors. So what kind of specific features that could give you is called graph modes that comes at different frequency. And as you can see in the image, it actually show you like at different frequency, the signal change at different frequency, and it actually show you the signal change at different frequency. For instance, from the low frequency, the signal are just highly clustered, and the patterns are highly clustered, and as you go to higher accuracy, even the neighboring pixels show different signal. So actually this type of tool has already been used in brain imaging, and also provided very useful insights of brain organization. So what are exactly the steps that if we want to apply this tool? So you need to start from the brain graph as we introduced in the first part. So here I show you an example of a connectivity matrix from diffusion tractography. And next is we need to define the graph signal. For instance, that means like at each node we need a brain response. For instance, fMR signal during a task. During a cognitive task. After that, we can directly using the graph Fourier transform to convert the data from the graph spatial domain to the spatial domain. Once we have the results, we can start looking at the specific property at each frequency. For instance, in this paper of Medaglia. So, for instance, in this paper of Medaglia, he used graph Fourier transform to separate like two frequency during two different frequency of the signal distribution. at a cognitive switch task. One is called aligned, which is at low frequency, and another one is called Libra. It's more like high frequency, at high frequency. And what's more is he found that only the high frequency signal was significantly correlated with the switch cost during a cognitive switch task, which is pretty cool, right? So once we have this nice tool, next is next what we can do. So we would like to apply for graph convolution in deep neural networks. So the key step of graph convolution is to use graph filters, which was so what the filters is a notion in natural image processing. That we have an image defined as we have an image defined here, and we use a we use a filter as a weighted sum by we have a sliding as a sliding weighted sum, as going from the left to from the first pixel to the last pixel in the image. So, but in the graph in the graph data, we couldn't do that because, and in the graph, the filters was defined with the help of the graph Fourier transform. So the key difference between the two notion of convolution is that in the spatial convolution, each filter we simply know it's a spatial. We can simply put the weights at each pixel. The filters was directly applied to its spatial neighbors because we already know from the imaging grid data. But in the graph convolution, the filters need to, in order to apply the graph filters, we need the neighbors are actually defined based on the adjacency matrix, which will tell you how these nodes are connected to each other. There is actually, the graph convolution networks has been one of the trend in deep learning in deep learning field, and there is various types of graph convolution have been proposed. So for instance, the one that directly apply the spatial directly apply the spatial spatial decomposition of the graph Laplacian, which is called the spatial GCM. And more widely used are the spatial GCM, which is actually using the neighboring information from the graph. The reason of using this is because for a large scale graph, it's a very computational cost, very high is computational. The computations are very high cost if you want to do the decomposition based on a large scale graph. So here I will mainly focus on two types of this spatial GCN. One is called ChapNet, another is first ChapNet. One is called ChapNet, another is the first order GCN. So in the ChapNet, so reminder again, both of these two types of network is based on the graph Fourier transform, just different graph filters. So in ChapNet, so in ChapNet, the filters are defined. The graph filters are defined by with the help of Chebyshev binomial expansion, which can be simply calculated using this formula. This formula here. The nice tool of this was nice with this by controlling the order of the key in the ShepNet, we actually could control the scale of information propagation in the graph. And a simplified version of that is called first order GCN, which is only take k as 1, and as you can see here, the graph features was actually simplified, could be simplified as just using the matrix multiplication, which can be calculated very efficiently in the GPU. So graph convolution networks has been started to applied in also in brain imaging. So here is there is studies like using GCN to isolate the one brain region, or it can be used to predict the disease for a disease prediction by treating each subject as a node in the graph and predict whether the subject has some disease, for instance autism, whether the subject has the symptoms of one specific disease, for instance autism. And in this talk we actually specifically focus on the application of using GCN for brain decoding. So in brain decoding, the input will be like a short time series of fMRI volumes, and then we extract the signals from each brain region and put it into the decoding model, and the model will tell you what type of the function the participant was performed in scanner. So let's dig in with the brain decoding and have fun. So brain decoding, the goal of the brain decoding is to actually infer the brain functions directly from brain imaging, to infer brain functions directly from brain imaging. And here we specifically focus on using task fMR, but there are other types of the data you can use, for instance EEG and MEG. So in the earlier, about 1 decade ago, the idea of brain decoding was distinguish a visual stimuli proposed by Haxby about 1 decade ago. So the participant need to viewing like different types of the image inside of the scanner, and then we need to train a classifier to distinguish those different types of images. And now on, the field of brain decoding has growing so fast, and people start to use more naturalistic stimuli, for instance movies, for instance using brain signal to reconstruct the frames from movies. So here what we are trying to do is because a previous study are mainly focused on a small population, for instance 7 to 12 subjects, but the generalizability of the tool, the analysis generalizability of the decoding model will be questioned if we applied it to more like larger scale brain imaging. But it's more challenging. So it became more challenging if we want to apply this decoding to a large scale brain imaging data. So here we, instead of using the linear SVM, linear tools, for instance, linear SVM or linear regression model, as shown in these two examples, we are trying to use more complex and nonlinear tools, for instance, using the deep neural networks. The benefit of that is we can actually generalize the model to not only to large populations, but also to different cognitive domains. For instance, working memory or language. So in order to make this happen, we need a large-scale neuroimaging data. So here we are actually using the data, the task ephemeral data from the Human Connectome project, which consists of over 1,000 healthy subjects and has been going through over 20 conditions. Over 20 conditions. By using the graph convolution, how can we perform the brain decoding? So we will start with so we need to start with one brain graph and the fMRI signal at each node. Next, we need to using the event data, using the event design data to extract the task, the signals corresponding to the task block, and then we input into the graph convolution layers, which will propagate the brain dynamics towards its neighbors. And to make sure the model learns various different types of the kernels, various different types of the kernels, we use multiple channels. And more complex information integration, in order to make more complex information integration, we use multiple GCN layers. So in the end, our model consists of 6 graph convolution layers and followed by 2 fully connected layers. And after that, using software methods to predict the task labels. The first example is to use the simplified version of first order GCN, and here is first order GCN, and which can be constructed by simply which can be using first order GCN. And despite its simplicity, we actually find the results are quite impressive. Here we show only using a 10-second window of fMRI data, the 21 cognitive states can be classified with accuracy as high as 89%. And compared to that, if we are using a linear model, we only achieved around 63% of accuracy. If we want to understand what was actually happening inside of the GCN, here we're using a tool called TSNE in order to visualize the representation of the features that has been learned inside of the GCN. So as the learning curve, so comparing to the raw data, which is highly mixed around different types of the motor, different types of motor task movements, different types of movements here. As the training curve reached as high as 19%, we can see the patterns, the 5 types of the movements are start to highly segregated from each other and became more clustered together to the same types of the movement, and to this, and also distinguished between left and right movement. And also we want another concern is we also want the model actually use biological meaningful features, but not biological features. In order to show that, in order to prove that, we use a tool called saliency map. So the saliency actually measures the importance of the input features by simply evaluate the change, how the change in the input features will affect the output prediction. So here I show you one result from the working memory task with the 0 back. It was simply recognition of different types of the image. So with the so we can see that the regions corresponding to the place image and face image are highly different. By place image is more concentrated. The features for place image are more concentrated in the median side of the ventral temporal cortex, while the regions for the face image are located more lateral side, which is surprisingly, actually very consistent with the literature and visual stimuli, with the area identified as PPA and FFA, specifically for recognition of the place image and the face image. And what's more, by looking at the confusion matrix, also found that the place image and face image, they nearly have no misclassification. They nearly have no cases of misclassification. All almost perfectly separated from each other. And the second example is actually to use the more complex model called ChipNet. So in ChipNet, we can actually, as I explained before, we actually could control the order k to control. We can actually use the variable k to control the scope of information propagation. When we only using when k is zero, it's only a global scaling factor by only considering each node itself, the information of each node itself. When k equals 1, when we use the first order, when k equals 1, which is similar to the first order GCN I introduced before, the information was only integrated from the direct neighbors, which means we only simply consider the one-step neighbors. And when we consider high-order neighbors, and high-order neighbors can also be considered by increasing the value of the k. So here we show two examples of using. So this is the results if we use by changing the value of k, how the decoding performance will change. So from the figure, we could see the first order have quite low performance, have the low performance, the performance lowest performance, and as the k increased to 5 or even higher, the performance all converged to a same level of performance, converged to a similar level. When k is 1, the model showed the lowest performance, and as the k increased to 5 or even higher, the performance all converged to a similar level. And what's more, and another interesting factor is we see like the effect of the k order actually have a different impact on different cognitive tasks. For instance, for the motor task, which we know that was mainly involved the primary, the motor and the sensory motor cortex, which is mostly just involved the motor sensory cortex, sensory motor cortex. And we see there is actually no significant impact of the K order. But for other tasks, for instance, working memory, that was actually involved multiple different networks and multiple different component functional modules and networks. We can see by simply increasing the K, the performance can increase from 88 to over 19%. So it showed us that so these results actually show us there is actually indicate there is different types of functional segregation and integration in the cognitive task. So these results also indicate there is various types of functional segregation and integration in the cognitive task. In order to show more detail what's really happening, in order to detailedly show the impact of the K, we did a similarity analysis on the representations using CKA. So what CKA simply tell you is by comparing the how similar the activations how similar of the activations between different layers or even between different models. So here we see when K as for the motor task, even when K is only 1, when K equals 1, we already capture very stable representation. But when we increase the K to high order, for instance 5, the representations are highly duplicated. Basically just to show you 2 blocks of different representations. And by comparing the similarity between the 2 models, we see that the K5 model only capture the first layer features and the 6th layer features, while we only using 2 layers to capture the low level representations and high level representations. But when we check the working memory task, which is more functionally integrated, which is integrated among multiple networks, we see a different pattern. When K equals 1, there is the blocks. There is multiple blocks inside of. There was multiple blocks that was actually learning totally different representations, with a very low transition between the layers. And as the K order increased to 5, the pattern became more stable, and when we're comparing the similarity between the 2 models, we see the K1 or the first order model actually capture both the middle level representations, only capture the middle level representations in the 6th layer, which means it is not enough. We require higher order model. Next, we also investigated how the definition of the brain graph will impact the results. By definition, so here, which includes 2 parts. One is the choice of different brain areas and different connectivity. Brain connectivity. So we start by using different brain graphs. For instance, the spatial graph, which is only consider the spatial adjacency of the shared vertex in the surface, and the anatomical graph, which is defined based on diffusion tractography, and functional graph, which is defined based on the resting state fMR, and the structural graph, which is constructed using the structural covariance. So first, the results actually show us once we have a high order key, the result. Once we have a high order GCN, we actually high order model. The definition of the graph in itself didn't have a significant impact, but still we can see the functional and anatomic, the functional and diffusion graph show the highest accuracy, and followed by the spatial graph, and the lowest is the structural graph. And also from the graph itself, we could also see a more like a locally efficient, a local connectivity. A local efficiency, more like a local, high local clustering, high clustering efficiency with a lot of like local connections. And in the anatomical graph, there is start to have more long distance connections across different modules. And for a functional graph, the long distance connectivity, the property of the long distance connectivity increase a lot, not only within each hemisphere, but also across between the two hemispheres. And by once we get the structural graph, we see a very significant randomized effect. And also we test by choosing different atlas what how it will impact the results. Here, and the basic conclusion is that the modularity that we use to identify the graph doesn't affect much, and actually it's the scale of the graph, means like how many nodes you have in the graph. Next, we also visualized visualize what type of the representation was learned inside of the GCN, inside of the ChipNet. So here I use 4 different tools, but if you see the picture, if you feel like the picture is too much, just focus on the TSN in the second column. So comparison to the raw data, which is highly mixed among different types of the motor task. We have the graph representation from the functional graph, which is highly separated for the 5 types of movements. But if we're using the spatial graph, we still have a very high decoding accuracy, but representation was not as distinct. Doesn't have very as high distinct as the functional graph. And when we do the same analysis for the working memory task, again for the raw data, it's highly mixed among the 8 types of the visual working memory. And when we use a functional graph, the 8 types of working memory, they were highly separate from each other, only have very small portion. We only have a random dot that jumps over another category. But when we're using a spatial graph, this mixing factors became more obvious, and the decoding accuracy also decreased. So that's basically the talk of the today. And next, I would like you to move on for the hands-on section, and you can find notebooks for the talk today, either from the GitHub repo, or you can you can use the Binder projects if you don't want to construct your own environment. And so the environment has already been prepared inside of this project. This Binder project, and inside of the notebook, there will be 2 main types of the practice. One is using the graph Laplacian and GFT, the more traditional graph theory tools from graph theory, including the graph Laplacian and graph Fourier transform. And the second part is using the GCN for brain decoding. And here we are actually, it was based on the PyTorch. It was based on the PyTorch 1 version, and you can start to have fun now. Thank you.